All NEET aspirants, you are listening to the podcast about me for NEET by S. Ramakrishna. In this podcast, the topic photosynthesis, where ATP, NADPH are used. In light phase, that is photochemical phase, the products formed are ATP, NADPH, O2. O2 is only the byproduct, one of the net product formed in light phase. That O2 diffused out of chloroplast and intercellular space of mesophyll into atmosphere. So remaining products ATP, NADPH are utilized in dark phase to reduce carbon dioxide to carbohydrate in this biosynthetic pathway. Apart from ATP, NADPH in dark phase, the other which are used are carbon dioxide and water. Next. The dark phase do not dependent on light but dependent on light phase products. How it could be verified? Immediately after light become unavailable, dark phase event continues to occur for some time till light phase products are utilized. Using the dark phase word is a misnomer, not at all. Dark phase should not be construed to consider it as it occur only in the dark, that is wrong. Do not dependent on the light, that is wrong. It dependent on light indirectly, it dependent on light phase products directly. So dark phase is independent of light, dependent on light phase products. Next. Melvin Kelvin and co-workers, they discovered a pathway, the carbon dioxide fixation by using chlorella, unicellular non-motile green alga that belongs to chlorophyceae. They used C14 radioisotope to trace the events in case of the biosynthetic pathway. So initially they discovered that a 3-carbon organic acid formed by CO2 fixation. This 3-carbon organic acid is 3-phosphoglyceric acid. In short, it is called phosphoglyceric acid. Next. Scientists investigated in large number of plants whether 3-carbon acid, the PGI, is the product in all plants or is any other compound formed due to carbon dioxide fixation. They noticed that in some plants, maize, sugarcane, sorghum, the initial carboxylation product, it was a 4-carbon compound, that is organic acid, oxaloacetic acid. So the plants in which first carboxylation product is 3-carbon called C3 plant. The plants in which first carboxylation product is C4, that is called C4 plant. Whether C3 plant or C4 plant or some other plant like succulents, crustacean acid metabolic pathway carried out in case of succulent. So C3 plant, C4 plant or CAM plant. The C3 pathway is only the pathway for biosynthetic pathway for making varieties food materials. Next, what is primary CO2 acceptor? Scientists struggled a lot to understand the dark phase reactions for a long time. How many carbon atoms would a molecule have after accepting the carbon dioxide? It would be 3-carbon, that is phosphoglyceric acid. To their surprise, accidentally, the serendipitous discovery was that there was a 5-carbon ketose sugar instead of 2-carbon. The 5-carbon ketose sugar was ribulose 1,5 biphosphate RUBP is the primary acceptor. And it is a surprise, RUBP, a 5-carbon compound, and after accepting it, how the 3-carbon compound is formed. Scientists, after investigation of so many number of years to know about what is the 2-carbon compound, but accidentally they discovered a 5-carbon compound. They spent the time to discover the 2-carbon compound, but accidentally they discovered a 5-carbon ketose sugar. So RUBP, a 5-carbon ketose sugar, after accepting one molecule of carbon dioxide, 5 carbon, 1 carbon, it is a 6 carbon. So two molecules of phosphoglyceric acid formed. 2 molecules of 3-PGA formed. Next, in case of Kelvin cycle, the Kelvin and co-workers, they discovered the pathway, biosynthetic pathway. They called it as a cyclic pathway because there is a regeneration of qualities RUBP. That's why it is a cyclic pathway. It is called C3 cycle, Kelvin cycle, and also carbon reduction pathway. Otherwise, photosynthetic carbon reduction PCR pathway. So C3 pathway, Kelvin pathway, PCR pathway, and carbon reduction pathway. Next, in Kelvin cycle, there are 3 phases: carboxylation, reduction, and regeneration. Carboxylation of RUBP, reduction of PGA, regeneration of RUBP. The first step, carboxylation. It is a passive step because neither ATP nor NADPH2 utilized in case of the carboxylation. It is the most crucial step. Why it is the most crucial step? Because RUBP undergoes carboxylation by accepting the CO2 catalyzed by the enzyme Rubisco, ribulose 1,5 biphosphate carboxylase oxygenase. It acts as carboxylase as well as oxygenase. So the carboxylation, proceeding for carboxylation is most crucial step because it is a dual role, carboxylase as well as oxygenase. So one molecule of RUBP, after accepting one molecule of carbon dioxide but utilizing one molecule of water in presence of Rubisco, and it formed 2 molecules of phosphoglyceric acid, an intermediate compound. Why it is an intermediate compound? As the reaction occurs, why you are calling it the intermediate compound? Because first reaction it is RUBP plus CO2 and PGA formed. Then G3P is going to be formed. Again it is RUBP formed. So in Kelvin cycle perspective, it is intermediate compound. In carboxylation perspective, it is initial carboxylation product, that is PGA. So a stable intermediate PGA formed. Next, reduction. In this reduction, in case of several steps of this reduction, in this finally carbohydrate is formed. So PGA got reduced to G3P. From the G3P is the precursor for the formation of already starch, later converted into sucrose. During this phase, 2 ATP, 2 NADPH are utilized in the reduction of PGA to glyceraldehyde 3-phosphate, G3P, otherwise it is called 3-phosphoglyceraldehyde. So 2 ATP, 2 NADH, 2 NADPH are utilized. So in this carbohydrate formation occur. So 1 carbon dioxide, 2 ATP, 2 NADH. So for 6 turns of Kelvin cycle has to occur for the formation of it is hexose. So for 6 turns of the Kelvin cycle in reduction phase, 6 into 2, that is 12 ATP are utilized. 6 into 2, 12 NADPH are utilized for 6 turns of the Kelvin cycle. For 1 turn of Kelvin cycle, 2 ATP, 2 NADPH. So 2 molecules of G3P formed. G3P is a 3 carbon compound. Next, in case of regeneration phase, so to ensure the continuity of the cycle uninterrupted, RUBP has to be regenerated. It is a crucial step. Without RUBP regeneration, Rubisco do not act as carboxylase or do not act as oxygenase. So main substrate for Rubisco it is RUBP. So G3P converted to what it is RUBP. So in case of G3P, as it converted to RUBP, a 1 molecule of ATP utilized in the formation of what it is 1 molecule of RUBP. So for 6 molecules of RUBP, how many ATP are utilized? 6 molecules of ATP are utilized. ATP to NADPH. So 12 ATP, 12 NADPH invested at reduction phase, 6 ATP invested at regeneration phase of RUBP. Next, 6 CO2 in, 1 hexose out, 18 ATP in, 18 ADP out, and 12 NADPH in, 12 NADP out. So, for 6 turns of the Kelvin cycle are required for the formation of hexose. 18 ATP, 12 NADPH considered as assimilatory power. 3 ATP, 2 NADPH are considered as assimilatory unit. So, how many assimilatory units are required for the formation of an hexose? 6 assimilatory units are required. What is assimilatory power? 3 ATP, 2 NADPH. What is assimilatory? It is unit. Assimilatory power 18 ATP, 12 NADPH. So, at which step the ATP utilized reduction as well as regeneration? At which step the product of right phase NADPH utilized in case of reduction phase? I am signing off from this episode, and in next episode you'll see the C4 pathway.